my pleasure to welcome Dr. Matías Muñoz right now from Oklahoma State University in US. And so Matías will present his work on how birds communicate effectively with mismatched auditory filters. Matías, you can start sharing your presentation, and the floor is yours.
I think you should be able to see it now.
Yeah, we see it.
Yeah,
Perfect.
Perfect.
Thanks,
So
Matías.
thank you for having me. I will be talking about birds and about their vocal communication and their hearing. And in biology, there are loads of optimality models, right? We heard some regarding the movement of the cheetahs this morning. And in sensory ecology, we also have one, and this is called the matched filter hypothesis, which predicts or tries to make a relationship between the signals that animals make and the properties of their sensory systems. So if we were to have, for example, a signaler here in orange, which has a signal with a certain amplitude and frequency profile, and we have a receiver here on the other side on blue, the matched filter hypothesis predicts an optimal and perfect relationship between these two capacities, right? Signaling and perception. And this would be advantageous in, for example, noisy environments or in environments that are highly attenuating. And this was the idea proposed by Kapranica and Moffat. And this article begins, the first thing that it says is that animal sound communication involves a cooperative agreement between the sender and the receiver. And this is a quote that has stuck with me for a while now. So, and I think we will come back to this at the end. And I am showing frogs. It's not random because we have studies that show, for example, here in the y-axis, we have the frequency of the call of the frog. And on the x-axis, we have the frequency at which their hearing is better. So it's a relationship between the peak frequency of the vocalizations and the best hearing frequency of audition. And if we do this for a bunch of frogs, we find this very nice linear relationship, very close to a 1-1 line. So it's a isometric, right? Frogs hear with a peak best hearing that matches very closely the peak frequency of their calls. And that's why the authors say that this matching is especially impressive in frogs. And of course, frogs are excellent animals for all of us interested in animal sounds and bioacoustics. But there are other animals that are equally exciting to study bioacoustics, and those are birds. Birds have excellent vocal capacities, largely because of their vocal organ, the syrinx, which is highly effective, exquisitely innervated in some cases, and it's something that it's unique to birds. So we wondered what happens between this relationship between how an animal hears and how an animal vocalizes in birds. And I will show you, I will play back a sound. I hope you can hear me. You can hear it. I will play this 30 seconds of a mix of different sounds made by birds. So that's sort of the diversity that we are talking about when it comes to the study of bird sounds. And macroevolutionary studies of the sounds made by birds, by songs or calls or all sorts of vocalizations, we have plenty of those. For decades, people have been studying comparatively the vocalizations of birds, but we don't really have a good understanding about the matching between hearing and the vocal structure of birds' vocalizations. So what we did in the methodology is that we looked into the literature to collect auditory data, which are called audiograms, which are these figures that have frequency on one axis on the x-axis and threshold, auditory threshold on the other axis. And they depict these auditory curves. Here, there are two different methods of creating these auditory curves shown by auditory brainstem responses and by psychoacoustics. And essentially, this curve shows you the frequencies at which animals hear best, or below this line, for example, they don't really hear. It's too low for them to hear, and they hear everything sort of that it's above them. And the lowest point of these curves, here at 3 kilohertz for this species, that's the most sensitive point for the species. So we collected this data from the literature, and we also, for those species that we had auditory data, we collected audio files from repositories like Xenocanto or the Macaulay library and analyzed them with Raven and R. And we obtained data for 72 species of birds. This is pretty much all there is in terms of audiograms for birds. We only collected for adults, and this is the distribution of in the phylogenetic tree. So you can already see that there are some groups. These are different orders that have been more extensively studied. Passeriformes, for example, the songbirds, because well, most of the birds are actually passeriformes. And to a lesser degree, the owls, strigiformes, the galliformes, the chickens and the fowls, and the ducks. And our data looks like this. In this figure, we have two lines. We will start with the black one, which shows the power spectrum of the vocalization. This is simply a way of representing sound that decomposes it in terms of amplitude and frequency. And we were interested in this peak frequency, right? The frequency at which the amplitude is highest in the vocalization. Or other way to say it is the frequency with the highest energy content in a call. And in orange, we have the auditory curve of this species. This is a falcon. And we collected also this, and we can see the type of data that we obtained. And of course, when we do this large macroevolutionary analysis, we find diversity. First, this is how it looks for some of the species that we studied, the relationship between the spectrum of the vocalizations and the auditory curve. So the first thing that we measured from this data, first, from the vocalizations, we measured the peak frequency. Again, it's the frequency with the highest energy content. In this case of this sample, it's like 5 kilohertz. And we related the frequency against body mass. And what we find is this negative relationship between mass and frequency. Different colors are different orders. And this is something very well studied. This is something that we know for pretty much all the vertebrates. Larger animals tend to produce lower frequencies. When we do the same, but for the audiograms, here we measure the best hearing frequency, which is the lowest point of the auditory curve, which is the one to which they are most sensitive. And we relate it to body mass. We also find a negative relationship between the frequency of best hearing and the body mass. Interestingly, these two curves have different slopes. So I will plot here both of them together. In violet, we have the vocalization peak frequency, and in orange, we have the auditory tuning, which decreases, but with a much less pronounced slope. There are implications for this. We, for example, would expect a perfect matching between vocalizations and hearing for birds that are around mid-size. For birds that are small, we would expect vocalizations to be higher frequencies than the best hearing frequency. And on the opposite end, for birds that are larger, we would expect their vocalizations to be lower relative to their most sensitive point in hearing. So one of the implications of these results is that body size evolution will have a larger impact on vocalizations than on audition. So if body size changes for whatever reason along the evolutionary history of a bird group, then this will have a larger impact on the calls than on their audition. And this is one example of a group of birds, the galliformes, with a very large variation in body size, right? From ones that weigh 40 grams to the big turkey that weighs almost 6 kilograms. Next, we related these two variables, peak frequency versus the best hearing frequency, and we didn't find any relationship between these two. This figure is equivalent to the frog figure that I showed you at the beginning. So this is a clear difference with what we found for what other people found for frogs. In birds, we cannot predict the peak frequency of their vocalizations based on knowing the best hearing frequency. And when one reads the animal communication literature, we will find that a lot of times researchers mention that signalers and perceivers co-evolve. And when we think about coevolution, we usually think of these beautiful examples of pollinators and plants. So for example, Darwin observed this flower from Madagascar, and simply by observing the flower, he was able to predict the existence of a pollinator with a very long proboscis, a very clear example of tight and strong coevolution. Birds are not an example of such coevolutionary dynamic. Next, what we did was to combine both sources of data. So before we just analyze them by separate, the auditory and the vocal, but we can combine them and get these different metrics of the matching between vocalizations and audition. So we computed this delta frequency, which is simply the difference between the peak frequency of audition, which is symbolized by this little white ball, and the peak frequency of the vocalizations, which is this other ball. So in this case, it would be around 3 kilohertz, where vocalizations are higher than hearing. And when we do this for all the birds in our sample, we find that the delta frequency is very nicely centered around 0. And at first glance, we might think that birds are super finely matched, but when we look in detail, we see that there is a lot of variation, right? So it's not 0 because all birds are around 0. It's 0 because it balances out between those birds that vocalize with higher frequency than the best hearing frequency and those birds that vocalize at lower frequencies than their hearing. So the mismatches that we measured in terms of frequency, they are widespread. Another measure that we obtained was delta amplitude, which is simply a measure of how far in terms of amplitude is the sensitivity of the bird to the peak frequency of the vocalization. So the peak frequency of the vocalization is at 5 kilohertz in this species. We predict that this delta amplitude will be around 12 decibels, okay? It's simply a measure of mismatch or match in terms of amplitude. The lower the value of delta amplitude, the better the matching. And when we do this for all the birds, we find that delta amplitude of approximately for most of the birds is below 6 dB. When we look at the details, we see that there is of course variation, but it seems that most of the birds, no matter the widespread frequency mismatches, they have still some decent sensitivity to the vocalizations. 6 dB is not the lowest that it can be, but it's not massive either loss of sensitivity. So what we conclude sort of from these results is that despite the frequency mismatches that we observe with birds, avian hearing remains adequate for vocalizations. In this figure, we have the mean phylogenetic threshold computed for all the birds, which is again the lowest point, is the most sensitive part of the audiogram. And what happens is that vocalizations have much narrower bandwidths than the audiograms. So birds can actually fit a wide diversity of narrow frequency vocalizations within their auditory bandwidth. And this is actually what we see when we plot here the histogram of frequencies of the peak frequency of the vocalizations. We see this sort of match where birds produce peak frequencies that are correspondent with areas of highest sensitivity in hearing. So to start to wrap up, of course, birds must do more with their hearing than just communicate with adults. We just analyzed the vocalizations of adults, But parent-offspring communication is important in many birds, in most of them perhaps. And in many cases, the chicks, the babies, are even more vocal than the adults, like for example in the ostrich, where the babies are much more talking than the adults. Of course, other things that some birds do is that they can use their hearing to navigate. There is some evidence that albatrosses use infrasonic cues to navigate on the sea. And of course, birds must also avoid getting eaten. And in some cases also they use their hearing for eating. So there is a lot of ecological situations in which hearing needs to be used for birds. And that's why a hearing that is tuned only to conspecific might not necessarily be advantageous in a real ecological context where animals have to do many other things than just listen to conspecifics. So final remarks, birds do not adhere to the match filter hypothesis or to the predictions of the match filter hypothesis. This doesn't mean that the relationship between receivers and signalers is deficient. It's simply adequate for what they do. And I do not think that this is goes against the cooperative agreement of Capranica and Moffat. I think this just shows that there are different ways in which animals can fulfill this agreement. And I would like to thank my PhD supervisor and the organizers. And if you are interested, you can also read the preprint, which is posted in bioRxiv. Thank you.
Thanks a lot, Matías. Very nice talk. We already have some question on the chat, so like I will shoot you with it. We have Bastorma Forever who is asking, does the carrier frequency always make the frogs grow those neck bubbles? Sorry. The picture is of the frog puffing their necks to make those bubbles.
Sorry, say again, the carrier frequency of the frog?
Does the carrier frequency always make the frogs grow those neck bubbles?
I don't understand the last part. The frog?
The frog grow those neck bubbles.
Oh, maybe I can read it
maybe
myself from...
Or maybe I can ask Pastor Mata Forever to reformulate the question, because
okay,
I must say that I am not too sure. Also,
no, I see. I read the question. So I think the person is asking about the vocal sac. So when frogs call, usually there is a vocal sac that inflates under their head, and most frogs have it. That's a clear signal that a frog is calling. In some species, it's less conspicuous. In others, it's massive. In others, it's much more less of a thing. But all of them sort of have a little inflation, I would say, of the vocal sac. Yeah, that's the bubble.
Okay.
Yeah, so that's a male trait in calling males, calling frogs, is that they have a vocal sac.
Okay, thanks for the answer, Pastor Mata. Hope it was what you were asking. So we also received some question from Lorkosha. So she say, thanks, Matías, for this talk. She's wondering, if you use the public app to collect the vocalization, such as a Merlin bird or another one.
Yeah, so I collected them by hand, actually, from Xenocanto. So I checked each species on Xenocanto and checked the high quality recordings, and I made selections by hand to make sure that we only analyze the signals with high signal to noise ratio and were devoid of other sounds on the background. So this was a manual analysis of vocalizations that I downloaded manually from Xenocanto.
Okay. And so she asked a second question, do you think that urban noise could blur the relationship you are trying to test? Birds are known for adjusting their songs when they are in a urban context.
Yeah. Well, I think when at this macroevolutionary scale, we sort of assume that all these influences are smaller than the macroevolutionary picture. So we do not take into consideration, for example, other sources of ecological variability. One is noise, but we could also wonder if there are differences between males and females, or we could think of other ecological contexts that might influence. We just, for example, do not have people who have measured audiograms that I know at least, in birds living in urban versus non-urban environments, right? So I don't think that we can with the data that we have actually addressed that. The same with the vocalizations, right? We work with the ones that are available in the repositories. And these are already biased because when people record birds, they try to get the cleanest recording possible. And when we analyze them, we also want the cleanest recordings possible. I am sure there are effects of urbanization. I just don't think that we have the resolution to find them.
Okay, I see. Because yeah, at the end, when you are making your recording anyway, you have the urban noise that are also on the recording, but it's just that you remove them in order to get something clean.
Yeah, we try to get the recordings that have the clear signals. So yeah, we didn't.
Yeah, okay, interesting. So like we don't have more question at the moment, but like I invite everyone to go on the Discord on the dedicated channel of Matías. Like if you have any more question, Matías, thanks again also to
Thanks
come.
to you.
Yeah, to come earlier and
No
replace
problem.
that. Very nice of you.
No problem. Bye. Thank you.
And